Sorry. Hi, everyone. Thank you for joining us today. We had a great talk last week from Ranjani to kick off this series, and this week's series is brought to you by myself. Those who don't know me, I'm Professor Derek Richard. I'm from Queensland University Technology. I've been in TRI, I guess, from almost the very start when QET moved in, and look, we've moved away through the academic field and into the commercial field. Our lab has 4 spin-out companies in total over the years, we've kind of delved from being completely academic to being commercial as well as academic. And one of the people key to this, and I'll introduce more later on, is Dr. Ros Wilson, who is the CEO of one of our companies, CARPE VITAE. And so what I'm going to do today is I'm just going to quickly tell you about some of the science that backs up the CARPE VITAE, the start of CARPE VITAE. And then Ros is going to tell you a bit about what it does to be a business. And I guess from the title of my talk, it's about the academic pathway to translation. And I think one of the most important lessons I've learned as I've moved through there is, we're academics, we are not commercialization people, and we should know when to ask for help. My number one lesson I give out to anyone who asks me is, don't try and be the CEO of an academic company. Bring in an expert to do that. And Ros will maybe explain to you why that is. So look, I'll just point out that we, both Ros and I am a co-founder and shareholder in CARPE VITAE. We're going to talk about some of the technology, and Ros is the CEO of CARPE VITAE Pharmaceuticals. My interest and the interest in my lab is really in the pathologies of aging. We're interested in why you age. We're interested in what drives the pathologies of aging, and they're really interested in how we can target those driving factors and then even target the disease once it forms. And so we cover everything from cancer all the way through to dementias, osteoporosis, and just aging in general. And aging is, and you all know out there, the aging is a major problem. It's the reason our healthcare system is blowing out. The budget on it is blowing out. And it's only going to get worse. We look globally in the US, Europe, Australia, China, the population is aging rapidly. Childbirth rates are generally falling around the world. And so the problem is only going to get longer. But as we age and we live longer because of medicines and better treatments and better diagnostics, etc., earlier interventions, we live longer, but we don't necessarily live longer without disease. So what we are doing basically is we're living longer, but with disease. And so disease is not killing us as it would do a way back. So why is disease more common when we age? Well, it is a kind of natural process, and most of you might be familiar with the hallmarks of cancer, but we also have the hallmarks of aging. And the particular hallmark that we're interested in our lab is genomic instability. Genomic instability is a pretty important hallmark. It drives many, many things. And as somebody gets older, you can actually see them getting the genetic code being mutated. So genomic aging is really important to us. In cancer, if you look at one of the universal hallmarks of cancer, it's also genomic instability. So our lab is basically a DNA repair lab. We're interested in keeping our genetic code stable and what drives instability. So, we know that cancer risk increases with age. If you're looking at one causal factor of cancer or the highest predictor of cancer risk, it's age, without a doubt. We obviously diet and other things have an impact on it, but it's age is ultimately the thing that drives cancer. And it does exactly the same in all mammals. And you can see here mice, after the reproductive prime, the onset of cancer is pretty rapid, or the risk of cancer is rapid. In the same case in humans, we look after children a bit longer, we live longer, and you can see here that that it really becomes exponential after the age of 40. So sorry for everyone in the audience who are over 40, but you're hitting that curve now, and it's not a good one to be in. But when we look at most of the diseases of aging, and when we look at dementia, Alzheimer's disease, cardiovascular disease, you can see really they all are diseases of aging. And you know, most of these diseases have a hallmark of genetic instability in there as well. That's at least part of the pathology, if not driving the pathology itself. And so we're kind of interested in what's driving those diseases. And so what we hope to do is, now, if you look at the average age in Australia just now, it's 82.7 years, I believe, or roughly 83 years. And if you look at the blue there, that's the lifespan without disease. And I might not have labeled this properly, but if you look at the blue section, that's life, healthy life. So if you look back at 1980s, man, say in Scotland, or women in Scotland, they would live to they get to about the age of 63. They would retire at the age of 65, and in general, they would pass away by the time they were about 67, 68 years old. And that's because they would die of diseases such as cancer or cardiovascular disease, etc. But now, obviously, we live a lot longer with disease. So we still get disease at the age of 63, but we now live to 82 years old on average. And we do that with the help of medications in modern medicine. And so scientists have come a long way making us live longer, but look, our health span has actually not increased in that period. So we're looking at genomic instability. In particular, we study premature aging syndrome. Here's a child at year 1, year 2, year 6, year 12, and year 32. This is a particularly long-lived aging syndrome that we're looking at here. By most of these aging syndromes, the children will die in their early to late teens of cardiovascular disease, stroke, dementia, cancer. So everything that you'd expect an old person to die with, they'll die with in their early teens. And all these aging syndromes are driven by defects in DNA repair genes or defects in proteins that cause genetic instability. And the progeria syndromes are great examples of that, but we have many other diseases of aging, all rare, but all driven by defects in DNA repair. And so, look, my story really starts a way back, and this might seem a little bit unusual. I'm a microbiologist, so, and I think there's many of us microbiologists swapped over in some point of our life to other areas of science. So I started off studying organisms that lived in volcanoes, organisms called the archaea, in particular, Sulforbus sulfataricus. This gentleman in the slide you can see here, if I can maybe point to him, is a gentleman called Patrick Forterre. He's really famous because he discovered topoisomerases. So anyway, these cancer biologists will be familiar with the topoisomerases. So he discovered them in the archaea, living in these boiling volcanic pools. The reason we studied the archaea there is really because they shared about nearly 2 billion years of evolution with human eukaryotes. So archaea and eukaryotes are very similar. Both are very distinct from bacteria. So we studied these microorganisms there. And that really led us to discover what we believe was kind of a pathway of cancer development. It led me from switching from archaea to humans when I really identified this new DNA repair pathway. And ultimately, it led us in a group led by Dr. Emma Boulderson within the Cancer and Aging Research Program to develop a lead compound that's currently going under medicinal chemistry optimization. And we called it DECO-1. Our lab always names drugs after our new technologies after the inventors. So the inventors are Derek, Emma, and Ken. And so that's where our names come from. We have another drug which has other inventors' initials on it. And so what does this drug do? So basically what we believe this new pathway does is when we hit about the age of 40, this pathway is triggered and it results in a downregulation of your DNA repair systems. And ultimately, that leads to a rapid increase in mutations per every year of your life. And those mutations are then driving diseases such as cancer and dementia. And so we managed to get a drug, and we switched the pathway off with the drug. For those that don't know, we have a $7 million drug screening facility up on level 6 of TRI. It's open to anyone at TRI who wants to use it. We launch and go open to everyone in the next month. So you're all welcome to come up and use the facility or work with us to use the facility. So we got over $7 million worth of equipment there. But anyway, so our lead compound that we identified increases the age of a worm called C. elegans by 20%. If you give the C. elegans a high stress environment, which causes damage to its genetic code, we will double the lifespan of that C. elegans. Increases the DNA repair capacity of human cells. It reverses osteoporosis in mice. Increases autophagy, which is associated with rejuvenation. It stops arthritis progress in mice. Here's just some of the images here, but so we can, in an osteoporosis style model in mice, we can add a drug and reverse the impact of osteoporosis, promotes autophagy, which is associated with juvenescence. And we can stop the progression of arthritis with the drug. And so it's all pretty exciting times for us. That's our drug treatment there. But this is the point when scientists have to kind of sign off and bring in help, because, you know, doing this on our own is not possible. I do science because I love science, and that's what I'm good at. I'm not a good CEO. I don't plan on being a CEO, and therefore I went out there. I did actually manage to raise seed funding, which then allowed me to hire a CEO. But now I hand over that highly skilled and highly technical part of the whole operation to the CEO. And Ros is now going to tell you about why you would do that. So Ros, you just tell me when to change slides, and I'll keep controlling it from the side.
Okay, thanks very much, Derek. And thanks for that great background to CARPE VITAE and what we're trying to achieve. So maybe just a couple of words about me before I get into the substance of this, just in case some of you are wondering why you should pay any attention to what I have to say, other than that hopefully I'm a reasonably engaging speaker. I studied medicine many years ago and then moved quite rapidly to the dark side and joined the pharmaceutical industry. And I was very fortunate to actually join the probably the Death Star equivalent of the pharmaceutical industry, which is Hoffmann La Roche, one of the world's leading pharmaceutical companies, where I really learned the ins and outs of drug development and drug marketing and commercialization, really to what I would call industry gold standard. And I've then been even more fortunate to come back to Australia and apply some of that experience and knowledge and tweak it as well, because of course we operate in a very different environment to a massive international global pharmaceutical company. Although I have to say that a lot of what I learned and certainly a lot of the discipline of the thinking and the approach is very, very transferable from the big pharma environment to the biotech startup. I've done a couple of other things, a bit of consulting. I was CEO of an ASX listed Australian company called Factor Therapeutics, which did some work in wound healing. And now with working with the team at CARPE VITAE, I'm taking on an early stage project, which is really great fun, and I'm excited about the prospects. So in terms of how I sort of approach involvement with a company like CARPE VITAE, really beginning with the goal, which is that we want to turn academic research into a commercial reality. But the first point I suppose I want to make is that commercial reality can mean different things to different people at different points in time. And so this slide is really to sort of demonstrate that. You have your kind of light bulb research that produces some intellectual property. That's a form of commercialization. Produces publications, which is another form of commercialization. Publications are a terrific way of obviously getting some awareness out there of the work that you're doing. You may then go on and out-license the technology to, and in exchange get to put some coins in your St Andrew's flag Scottish piggy bank. Or you may indeed then go down the route that Derek and his team have of establishing your own spin-out company. But I would argue that the real commercial reality is what we see in this photo with this lovely lady and this person standing next to her at the bedside, which is that commercial reality for me is about producing medicines that ultimately benefit patients. So go to the next slide, Derek. At the TRI, I guess this is kind of maybe my sort of expression of where the starting point has been for me with CARPE VITAE, which is, hooray, we've got some seed funding, what happens next? So if we take a look at where the TRI sits in the continuum of drug development, this diagram is showing two things really. The two sort of series of coloured boxes at the bottom there. The top is development of a typical pharmaceutical drug. The bottom is development of a companion diagnostic. They're slightly different. If we have time, I'll go into that, but just suffice to say that they do follow similar principles. And you can see that the period that involves the basic research, the discovery, and the preclinical development before anything goes into human trials can take anywhere in the order of 3 to 6 years. And then beyond that, clinical trial phase, 6 to 7 years, approval and launch of a product between 6 months and 2 years, depending on the pathway that you go down. So I always say that drug development is not for the faint-hearted. And in terms of the kind of model, I suppose, that we operate with here, with CARPE VITAE's thinking, and also I believe through the philosophy of the TRI, is that we can sort of take that excellent science, build on it, generate the preclinical proof of concept, potentially go through to the sort of first clinical translation trials as well, so that perhaps phase 1, even into phase 2. But at some point, we probably expect to hand the baton over to let Big Pharma then drive the technology further through the more costly mid and late stage trials period and the approval and launch process. So that's kind of one view of the journey. If we go to the next slide. I think the other thing to think about, and it kind of harks back to that photo that I showed first up, is that it's really important to begin with the end in mind. And what I mean by that is when we're looking at developing a new technology, whether that's a new medicine or a new medical device, it's really important to start as early as possible thinking about what patients and what customers are we trying to actually develop this product for. So just a little bit of kind of technical business language. Just bear with me 1 second. Just getting the background noise under control there. A market is defined as a group of customers who have a common need or a common interest. And so when we're thinking about developing new medicines or technologies, the question is really what need for your customers will you be satisfying? Or what customer pain are you relieving? And some of the best business ideas come from contemplating really the problems that need to be solved. And also then I think what that then sort of leads on to is the thinking there is to be a product for a market rather than a product that's looking for a market. So if you already have clarity around what it is that you're trying to, the end game that you're trying to get to and achieve with all of the research that's happening, that can, I think, provide a lot of clarity and good direction for the product. I've been involved in some products that were probably more of the case of a product looking for a market, and it can really be a struggle there to really define and convince even people, potential investors, collaborators, that it's going to be a worthwhile endeavor. And beyond that, and I think this is one of the great things about CARPE VITAE, not just be a product looking for a market, but be a portfolio of products, be more than one. One of the biggest challenges in the Australian biotech sector is that we have binary outcomes because companies get established with the spin out and the licensing of a single piece of technology. So if we look at the next slide, it's really the point that I'm making here, that and I suppose what happens is you raise money, there's a lot of excitement, you do the trial and it doesn't work, and suddenly you've got a bunch of unhappy investors and you're back to square one. Whereas if you look at the way that the well-funded companies operate, and you don't have to look at a Roche for this, there are some pretty reasonable examples in Australia, and certainly an awful lot of them in the US, that they may start with a single asset, but they work very diligently, not only progressing that asset, but also building out their portfolio so that you have what the investors and the bankers like to call multiple shots on goal. And that's, you know, simply a fact of the incredibly high attrition rate that we see through drug development. So if we move on to the next slide, Derek, the other lesson, I suppose, or to think about is building an approvable data package, even when you don't plan to be there at the end. So I know that a lot of people involved in biotech, not only here in Australia, but elsewhere as well, understand that, you know, we may not have the expertise or the interest even to become a company that has our own sales and marketing force and distribution channels to make a product and sell it directly. And so we're often planning that we would, you know, exit that technology at a point in time, and usually, let's say, you know, end of phase 1 or end of phase 2. But of course, the important thing there is that whoever decides to assist you in that exit and take that technology on is intending to take it further through and ultimately to approval and launch. And the closer the data package that we present to those people at that point in time to something that they would have done themselves, with their FDA approval in mind, for example, the more likely they will be to be interested. And that really speaks to understanding what is needed to fully manufacture, fully develop, and fully approve a product. And I'm going to spend a little bit of time going through that. But I thought it would be quite hopefully interesting to sort of just reflect for a couple of slides on what I think are kind of critical aspects of what this data package might comprise. So if we go to the next slide, Derek, the critical business tools, the first one is what I call a target product profile. That's a pretty specific terminology for drug development. You'll find medical devices get called different things like design inputs or user needs or critical performance characteristics. But the point is the same, that from as early on in the process as possible, think about what a clinically meaningful product will look like, so that when you are then evaluating results as the data builds, you have a benchmark against which to measure. I give a good example, and it's unfortunately an also common thing that we see, is that especially in the Australian biotech sector, is a company might come out. There was a company recently came out and said, oh, we finished our phase 3 trial and we didn't hit all the primary, we had co-primary endpoints and we didn't hit them, but we did find a benefit in a couple of subgroups. Now, I don't need to explain to scientists the perils of subgroup analysis and cherry picking of data, but really what happens there is that companies can, if they don't have clarity about what they're trying to achieve, then they can fall into the trap of trying to make the best of what they have, which might be a good reason to keep something limping along for a few more years. But at the end, if you have veered too far away from what you understood to be a meaningful product that would be approved, prescribed, reimbursed, and used, then very soon that will catch up with you, and you'll struggle when you're trying to license out or partner your technology. So it's a really useful benchmark. And the other thing that happens is it's a living document. So as the data in the field evolves as well, so for example, if a new preclinical model were to come out that sort of provides better definition or clarity on efficacy or safety of a particular technology, then you can incorporate that in. And similarly, if clinical trials that are coming out from competitors come in that tell you that these are the endpoints that you need to focus on, or actually you need to go for a higher level of benefit. So the target product profile is a really important document, and it's one of the things that we're going to be working on quite intensively with the CARPE VITAE technologies over the coming weeks. Development plan sort of falls out of that, really. In order to deliver what we've described in the target product profile, what research and development do we need to do? Not surprisingly, from what comes next is how much is it going to cost to deliver that, and how much is it going to cost to deliver all of that? You may not look to raise and probably won't be able to raise it all at once, but it's important to have an understanding of the scale that you're contemplating. and document the science. On what basis are we making these assessments? Making sure that publications are kept well sort of filed, presentations are all accurate, up to date, referenced. Just makes it an awful lot easier, especially when it comes to due diligence, to really see that the claims that the company is making about its technology are validated. Then we get to kind of critical people. A board is important for making sure that the business is managed overall in a competent way and in a way that meets the requirements of the Corporations Act, because when you set up a company, you become, the company is legally governed by the Corporations Act. But then you also need a hands-on management team, which is, in the case of CARPE VITAE, that's me, who's accountable to the board for the hands-on running of the business. And so my role within CARPE VITAE is really to liaise and I guess, you know, allows Derek and his team to focus on what they do best, which is the science, and I take care of all the other bits and pieces. And then scientific advisors to provide not only an external expert review and a bit of a sanity check, but also certainly when it comes to talking with potential investors, an external endorsement of the technology is also extremely helpful. Then the next kind of group of things is really about inputs that you need. And these are all of the work streams that go into producing a submission package that will be ultimately approvable by the FDA, the EMA, the TGA, you name it. So chemistry, manufacturing and controls, you know, that governs making sure that you can manufacture and supply to the necessary quality and scale, depending on your phase of development. Clinical expertise is in multiple aspects, and I've just listed a few there: designing trials, interpreting the results, but also very importantly, safety monitoring. Clinical pharmacology is also key for that sort of initial translation into the clinic to make sure that the starting dose and escalation plan for the first in human studies is based on the data that's been generated in the non-clinical setting and is going to meet with the market's needs. I should point out, these are not in order of how they feed into the process. I decided that I was just going to put them in as in alphabetical order, because actually I would argue that you need to be thinking about all of these things pretty much in parallel as things go. So commercial health economics, who's going to use the product, who's going to prescribe it, who they're going to prescribe it to, and who's going to pay for it, which of course increasingly becomes an issue. The preclinical expertise, of which we have plenty here within CARPE VITAE and of course within the TRI more widely. Quality is part of CMC, but I make the point there that when it comes to developing a medical device, that is an entirely quality-driven process. And it's really, really important to have all of the standards, the procedures, the documentation set up as early as possible. Just saves a whole lot of pain and backfilling down the track. And then finally, regulatory people who not only make sure that the data you've generated is going to meet their requirements, but actually help you interact with the regulators, because regulators really speak their own language. It's the value of having a good regulatory person advising you is incalculable. So those are kind of like my sort of, I think, what you must do. And then, of course, the big thing is money makes the world go around, even in scientific research and the altruistic pursuit of benefits for patients. I'm showing my age with the meme that I chose, but I couldn't resist. And really managing the expectations of our investors is also very important, and also particularly depending on the type of investors that you have. If you have somebody who understands biotech, it's a very different type of conversation to somebody who's maybe investing for philanthropic, and indeed sometimes in the case of seed investors, even personal reasons. And no motivation is the wrong one, but it's just important when you're working within the company to understand what the motivation is and ensure that expectations are being set appropriately. Also bearing in mind that usually even the most philanthropic of investors will be looking for some kind of financial return in a 3 to 5 year time frame. And the way that you assist your investors in achieving that goal is that you progress your drug, your portfolio, your company, because the further you move things along, the more you create value, which is then going to attract new investors, and allow your early investors, if they wish, to either take some profits or indeed to exit completely. So that's that. I mean, there's a whole other talk really in fundraising and financing for biotechs, which I won't go into now, but just sort of bearing in mind what to think about from a commercial or financial point of view. Which I think then brings me to my last slide of just kind of take-home messages. That commercializing great science is equals helping patients and not just filing a patent or getting a company number. Begin at the end. Who are your customers? What do they need? What pain are you going to relieve for them? Build a portfolio. I really don't like the cliche multiple shots on goal, but it is incredibly apt, and you will hear it if you get into the setting up companies and talking to potential investor scene. Start early with the non-science stuff. This is probably a plug, hire more people like me when you can. Get good business input early. Doesn't need to be a huge commitment. At the moment, I'm doing 3 days a week with CARPE VITAE. And that's, I think, pretty much right on track for where the company is right now. And something that I think scientists are really, really good at: be disciplined and be organized, because without a doubt, whether you're speaking to an investor, potential partner, a licensor, a pharma company, a prescriber, making a first impression, you get one chance to do that. And I think the more clear you are about what you are trying to achieve and how you plan to go about doing that, the better. So that's it for my sort of whistle-stop tour of commercialization. And back to Derek. You're on mute, Derek.
Yeah, thank you very much, Ros, and thank you for coming along. This is just this is the CARPE team up in level 6, so we're quite a large group, and we actually do have our own lab CEO who picks up new IP, identifies it, and pulls it through the first stages before spinning out in a company. So Rodney Kuzak is pretty key to that, and is great having him as part of the team. And then just some of the people that fund us, but we can stop there. If anybody wants to ask, we're going a little bit over time, but if anybody wants to ask any questions of either myself or Ros, you can see from Ros's talk that the whole process is quite daunting, and that's why I think it's pretty important that you get it right and get the experts in to help. Does anyone have any questions? No, you can, if you don't want to speak, you can send a message if you want, but.
Everybody's stunned.
Yeah, seriously. I did have a big picture of you, Emma, but I forgot, I obviously put up the wrong version of the talk and you weren't in it. No questions then? All right, well, I'll just maybe finish up there and thank TRI for organising the event, and for everyone coming along, and in particular for Rosalind Wilson, who gave a great talk on the multi-assets, or a whistle-stop tour of the multiple different things you have to consider when you're commercializing a product. So thank you, Ros.
And Derek, can I ask a question?
Yeah, sure. Yeah.
Ranjana here. Thanks for both of you, a really interesting talk. I was just wondering, when you showed the big group at the end, how do you manage the academic and the commercial side all in the one place?
So I guess we're pretty well defined. So all our commercial projects actually have an advisory committee within the university, and so they oversee the academic, they oversee the commercial aspects of the academic projects. QATs are reasonably liberal in its approach to this kind of merging of academic and commercial programs. Yeah, so it's basically you do your your due diligence or whatever. You put in the QAT puts in a panel in place just to oversee that everything is operating correctly and the best interests all parties has been met, and that's how we deal with it at QT. I'm not sure how UQ deals with it or any
I'm just
other.
wondering on a sort of week-to-week basis, do you have ideas coming up through the lab meetings that then you think about commercializing, or how does that kind of run for you?
Yeah, so everything academically goes through our own commercial project commercialization office. So it goes to QUT's main office. Rodney usually is the first port of call to somebody to work through the idea they have about their technology, what kind of addressable market it's going to do, unmet need, etc. And then if Rodney's pretty happy with it, he'll then go to QUT, disclose it to QUT, and then we work from there. Nothing automatically goes into any of our companies. It's all open, so anyone can come in and license anything that we develop. There's no automatic rights of CARPE VITAE or any of our other companies to have IP from the university. As Ros alluded to, CARPE VITAE is just licensed another technology from our lab. It's in negotiations with another technology from another institute, not QUT, as well. So it's not completely QUT centric, the organization. So it's open to IP from other regions of Australia, other institutes, etc. Ros, do you want to say anything on that, or I guess you're more?
Yeah, look, I think I think I've got a good relationship, and Rodney and I speak regularly, and really I suppose part of my remit is also to ensure that sort of that there's a good sort of delineation of the work that the Q team are doing for CARPE VITAE, the company, as distinct from the broader research program that they're pursuing. And so again, I think it helps to have, you know, to formalize some of that to a certain extent. So there are research and collaboration agreements in place, and I will be part of that management group. And then ultimately, you know, where QUT is continuing to develop assets that CARPE VITAE seeks to commercialize, then I essentially become the QUT group's customer. And I think Rodney is one of the key people for sort of being a contact for me in that sort of relationship.
So Derek, are people recruited to work in your lab with the understanding that they're doing potentially commercial work, or is that just they're just recruited as normal academic postdocs and then, you know, you educate them, or how does that work?
Yeah, so I guess most people in my lab have been around for a very long time, so we tend to hang on to good people and keep them around. Yeah, so you know, we do recruit in, and they so for instance, Emma's group, we have two large commercial projects in the lab. We have a number of smaller ones, but Dr. Emma Boulderson runs one of them, and that group sits in there, and she'll recruit in commercial postdocs or RAs to do the work in there. And then our other big group's led by Dr. Laura Croft, and that's on an oncology product in a drug company called CARPE Pharmaceuticals, or Repluca, sorry. And so, yeah, it depends. You know, I think both Laura and Emma will admit they've both gone through, along with me, very steep learning curves, because, you know, as academics, we're not actually trained in any of this. We're trained how to write papers and grants effectively and do good research. And the good research obviously is key to moving the commercial products. But yeah, no, it's, I personally think we need a learning curve. I do encourage learning in the team as well. We send people over when it was alive before COVID to Bio. There's a boot camp at Bio. We've had a number of people from the lab I've sponsored to go over there and do the boot camp, and then go on to do the next in the line. So there's two courses at the Bio. There's a boot camp, and then there's a more advanced course. So people have done both courses as well as local courses. And I myself, I went to MIT. I've done the Bio boot camps, etc., and I've gone to MIT and did a postgrad kind of course at MIT as well to kind of keep me up to speed. But that really just fills the gap. You know, that valley of death when you need the funding, etc. That kind of helps us fill that gap, and then but we still need people like Ros post that gap to come in and take control. You know, I think one important thing is, as scientists, we've got to think about a product. We're not very good at thinking of a product as a scientist. You know, we never have an ending question. We question everything. We go for perfection, and we'll go on forever for perfection. Whereas you need to think of a product. You need to draw a line at some point. Yeah. Any other questions? If not, we can obviously, I'm available by email, and if you want to speak to Ros, I can connect anybody from via Ros as well afterwards, or just come up and visit us. I'm on level 6, our lab's on level 6, and as I said, if you want to use any equipment, we have over $7 million worth of drug discovery equipment of various types up there on level 6. I'll maybe finish there then if no one else has got any more questions. So thank you once again for everyone coming, and I'm looking forward to next week's talk.